Very warm welcome to everybody. My name's Roy Anderson. I'm Director of the London Centre for Neglected Tropical Disease Research. And this is the first of 3 scientific roundtable discussions on reaching hat elimination, organised by the London Centre in collaboration with the HAT platform and the Royal Society of Tropical Medicine and Hygiene, and very kindly supported by Sanofi. It's obviously a timely event in the sense of that we've had 2 very difficult years with the COVID pandemic, and the number of meetings on HAT, of course, been quite significantly reduced. And also we've had the issue of continuing monitoring and evaluation of prevalence and infection levels during this rather difficult past 2 years. So the recent publications in this area, one in particular drawn to my attention on HAT, the elimination targets set by WHO for 2020, and the progress, the enormous progress actually, that has been made in trying to reach these targets. We need to always reflect that having a target which is elimination is very challenging for infectious diseases. It's always worth reminding ourselves that we've only eradicated 2 infections, that's rinderpest, as a veterinary infection, and of course smallpox, rinderpest in 2011, smallpox way back in 1980. So eradicating any infection is very challenging. Now, the objective of this set of scientific sessions is to identify gaps in research and knowledge that will help us to move towards the 2030 WHO goals of elimination of HAT. And we hope through these 3 sessions to be able to generate a list of possible research gaps which could be solved by appropriate support and appropriate interest from research groups throughout the world. And we look forward to coordinating research that arises out of these discussions. So a very warm thank you to the panelists who've agreed to participate in this meeting and given up their time for it, and particularly to Olaf Valverde and Florent Mabeau for their efforts in putting together the programme in collaboration with Catherine Forbes, who I wish to thank at the London Centre, who's done a lot of the organisation. Now, it's my pleasure to hand over to the chair for this first session, Gerardo Prioto from WHO, to introduce the panelists and start the discussion going. So, Gerardo, over to you.
Yes, good afternoon, everyone. Thank you for inviting me. It's an honor. I will try to keep up to it for moderating this session today. Maybe I will just make a few points of logistics for those that are attending. The first one I will say in French. Pour les francophones, il y a une traduction de tout ce qu'on dit, traduction simultanée en français. Pour les trouver, il faut aller tout en bas de l'écran dans Interpretation, c'est écrit Interprétation. Et là, vous pouvez cliquer sur le français si vous voulez écouter pour la traduction. Okay, I'm switching back to English. To say that if you have questions or comments to do, everyone can do it through the questions and answers box that you have also at the bottom of the screen. So please post there any questions you may have. And because we would like to end this meeting with ideas about research questions, gaps that we may have still in defining what are the important research questions to support the end of transmission drive. So, having said that, I will now introduce the panelists that we have today. Okay, I can see that in the chat already. It has been copied an introduction, the bio of myself. Okay, so I will just briefly say that I'm a medical doctor and epidemiologist, and I'm now working in the WHO in the team for HAT elimination. But prior to that, I worked some 10 years with MSF on humanitarian aid, and 10 years with Epicenter, based in France. But my first 2 years with Epicenter, I was based in Uganda, actually, and after I moved to France. And since 2010, I'm working with WHO. In the panel, we also have Professor Stephen Torr. So Steve Torr, he worked over 30 years on the behavior, ecology, and control of tsetse flies and mosquitoes. So he has a very large experience, and he is member of the UK's Tropical Development Research Institute, which is now called Natural Resources Institute, or NRI. And he was involved in many studies and projects using animal and artificial bites for controlling tsetse flies in many countries in sub-Saharan Africa. Also, he's very involved in developing simulation models and decision support systems for designing, implementing, and monitoring tsetse control operations. And since 2013, he's a professor in the NTV at the Liverpool School of Tropical Medicine and the University of Warwick as well. You have the full bio, or not full, but a larger bio posted to read. We also have in the panel Professor Philip Busher. So he has dedicated decades to the Institute of Tropical Medicine in Antwerp as a scientific researcher in human and animal African trips, mainly focusing on improving diagnosis. For 23 years, he was head of the unit of parasite diagnostics that developed new formats for parasitological, serological, and molecular diagnostics for African trypanosomiasis, also for visceral leishmaniasis and Chagas disease. This unit supported technically and scientifically many research projects of multiple groups, not only of the ITM. So we have enjoyed his support, technical and scientific, for designing many of our projects for research. He is now still active, even if he retired from that unit, active as a senior researcher in the ITM, and keeps assisting the HAT control and elimination program in the DRC in particular. We have also as panelist Deirdre Hollingsworth. I'm not sure how to pronounce the name. I think it's something like Deirdre. And sorry if I don't do it right. She is an epidemiologist who is using mathematical models and statistical analysis to study the evolution and transmission dynamics of infectious diseases. She leads the NTD Modeling Consortium. It's an international network of modelers. Her research foci are lymphatic filariasis, visceral leishmaniasis, and soil-transmitted helminths, other than the HAT. And we have also Dr. Wilfried Mutombo Kalonji. So Wilfried joined the DNDi in 2016, and now he's the clinical project leader and medical manager. And he has coordinated the fexinidazole clinical trials on behalf of the national control programs of the DRC in collaboration with the DNDi. He has over 10 years of field experience in human African trypanosomiasis research and case management. So this I try to make a briefer introduction of our panelists. And now we are going to pass on to questions and answers about the presentations that were already posted for you to see in advance. So I will briefly comment on these presentations, just as an introduction for the discussion. I have seen posted 10 presentations, and at least 8 of them, we are concerned today with at least 8 of them. if I consider that tomorrow is the day for the socio-anthropological issues. So I would comment that first I would take the one of the Institute of Tropical Medicine, where the presenter is Raquel Inocencio da Luz, because she approaches the issue of case finding, therefore the issue of diagnostics and screening tools, and looking at the situation where after this very strong decrease in the numbers of cases, we have a totally new situation, and we have lower numbers, but they are very spread. She's showing how spread they are in the map. And need to know that this is focusing on the DRC only. It's not a study looking at other countries. But it is an analysis pointing out to the need to adapt the case finding strategies. So, for example, she's pointing out passive screening should take a larger role, but active screening should still be there. And the question remains how to combine them better and how to do this in different settings, because there are different settings. And these settings that they are trying to categorize during the presentation include also the gray areas, they call it blind spots. So what are the strategies for blind spots as well? And there's also an important point made there about ensuring quality of the diagnostics done, because this is also a new issue we are having and going to have more and more with the disappearance, with the rarity of cases. Then connected to that, there's another presentation also by the ITM, it's Rian Smithers, looking at the economy, but looking at the economy at patient level, the family of the patient and the patient, what are they spending or what are they ready to spend? And there are some very interesting outcomes of their study, because they find that some patients are used to the situation. This is in the DRC, where they're used to waiting for the screening teams, the mass screening to come to the village. So in many cases, even if they should be seeking consultation because of feeling symptoms and so on, instead of traveling, they prefer to wait in the village for the screening team to come, which we have to take that seriously, because maybe not so many teams are going to be coming in the near future. Also, they are used to waiting for transportation and for food to be provided, because this is a situation that has been created by all the methods used for the clinical trials that have a special funding. So these are new situations and interesting points that we are not always aware of. So third, there is the DNDi presentation by Olaf Valverde, so which is showing clearly that the DNDi has had a very significant contribution for the improvement of the treatment of this disease that used to be terrible a few years ago. So, but I have to make one point about the history. When the historical part is presented, it is stated that DNDi developed the NECT study. And I wanted to comment on this because I'm in good position to say something about this, because I was personally involved in the NECT development. And I have to say, this is not quite correct, because the NECT study was developed by Epicenter together with NSF, not by DNDi. DNDi came after, but jumped on the moving train to help complete the studies and to do further studies to know how the treatment was working in children, in pregnant women, etc. About fexinidazole, it is stated how the simpler logistics are important, and also that we can avoid lumbar punctures for a certain number of patients, but not to all, unfortunately. This is important But it's also stated, and it has to be reminded, that fexinidazole is less effective than the former treatment, than NECT, and that can have also an impact. This connects with another presentation of the Swiss Tropical Institute by Mimi Das, and she presents interesting points via a modeling study where they are looking at the end of transmission. And they say, by introducing fexinidazole in particular, what can be the impact on the end of transmission? And it's very interesting because he's saying, on one side, fexinidazole could delay the end of transmission because it's less effective. So we may have more cases that are not correctly treated, they are not cured, and they remain as reservoir. On the other side, fexinidazole could help improving the access to treatment. And so maybe solving some of the situations of distance and logistics. So they're basically concluding that this potential delay could be offset by improving the case detection rates. But they have to be improved, and they say passive screening especially has to be significantly improved. I think they say 20% or so, to offset the negative impact of the introduction of fexinidazole. So it's a particular view. Personally, I think in that study they could have probably introduced some more factors that we know are happening around the dynamics of the end of transmission. That is extremely interesting. Then I have, okay, the presentation by Kat Rock on modeling. So she does a general description of what modeling has been doing, mathematical modeling. For example, that they can do test hypotheses, assess progress in the elimination, they can do predictions, and they can also simulate different scenarios by using modeling. Okay, but she points out clearly that all this modeling relies on data. So it's all quantitative data that can be obtained from different sources. And of course, I want to say, this is modelers, they are mathematicians, and they work on the data that they receive, and they don't have usually a presence in the field. So they rely a lot on the data and on what other experts may explain about this data. So it is all second-hand information, and that is why we always have to keep in mind that modeling is estimating things, but we shouldn't take it as the last word on a certain number of questions. Okay, so okay, this I already said. It was interesting also, she explains how modeling is a cycle of building the model, but then refining the model over time. So it's not the last result from the start. Okay, there's an interesting point that is made in that presentation, that to measure end of transmission, there are some limitations because the indicator is reported cases, but what happens when the screening doesn't have a good coverage? For example, how can models cover that aspect? And also the reported cases, there is a lag between the moment they were infected and the moment they are diagnosed, and that can be variable. So it's some of the limitations of measuring the end of transmission. Okay. And they can also, through modeling, go into health economics evaluations. Let's see, I think I have Okay, connected to this, because modeling is, of course, epidemiology. There are two presentations about the DRC by Professor Dr. Eric Mwamba, where it's interesting because they show the data of Dingila. This is very recent, where a team went to Dingila in the northeast of DRC. And they did an important effort to find cases, and they found 0. There were no cases. And so the question remains, they are asking, are there cases or not in Dingila? And so I want to connect that to modeling. Probably if we asked that question to modelers, they would have said yes, because there is an antecedent, recent still, of a large numbers of cases in Dingila. And so the models would have shown probably a lot of cases in Dingila, but the teams found 0. So it's important to contrast this. On the other hand, the other presentation about the Bangassou in the Central African Republic, a place where no cases have been reported for a long time. They are reviewing, reporting it. There is 21 cases that were happening in the lapse of a few years that were detected, sometimes indirectly, and that some of them may have been infected elsewhere. It's not clear, but some of them could only have been infected locally. So Bangassou seems to be a resurgence of cases. Okay, and the last but not least is the vector control presentation. This was done by Iñaki Tirados, and he takes us over the history of how vector control for tsetse flies evolved from insecticide ground spraying to the sequential spraying, taking into account the life cycle of the tsetse flies. And that was in the 70s. And but they had some problems, like the reinvasion of tsetse flies, and so they had to create barriers to try to block the reinvasion. But that took a lot of work, and the whole thing was quite expensive. Then there was the work done through with attractive odors for the flies that were identified, and that allowed to use artificial baits, which are those screens that can attract the flies. Also, okay, and that led finally to the tiny targets development. And tiny targets, it is explained how they are twice as effective as the other methods, and they have much lighter logistics, which allows for implementation. For cheaper implementation. These tiny targets are used right now in 6 countries, but they are always coupled with medical interventions. And they are also considering now building a strategy for reactive vector control in the new era of elimination. Also, animal bites can be used. There is an explanation of the technique and how that was developed. And there is finally a comparison in that presentation of the bite technology, comparing with spraying and showing that it's more cost effective than simply spraying. And also that it can be adopted or applied by the community members, and it can be either artificial bites or on cattle. But of course, all that has to be adapted to different settings, because there is no cattle, not enough cattle in many of the settings that need to be controlled. So that was as an introduction. Sorry if I took a long time. But now I will open to questions for the panelists. I know in the chat there was already posted some first questions that were sent before this meeting. Now I have to find them. Okay. So I have a first question is saying, what are the main gaps in hat research that need to be addressed to contribute to elimination? So that is a very large question that probably we will end up attempting to answer at the end of the 3 sessions. I would not throw that out right now. The second one is, what impact does vector migration and disease transmission and control have on the WHO goal of eliminating jihad by 2030? So that one is about migration of vectors, if I understand it well. And then what can be done about displaced populations, warm, torn, and poverty areas in terms of elimination? Okay, so why not? We can start with the one about vector migration. And may I ask Steve to comment on that question, please?
Thank you very much, Gerardo. I hope you can hear me. So I think, thank you for the question. And perhaps I think in the case of tsetse flies, it's more considering about their general movement. They don't seem to, as far as we can understand, they don't make long-range movements, like, for instance, black fly with river blindness, where those long-range movements have big epidemiological implications. For tsetse flies, being larger insects, they're not carried by the wind. They move under their own power, and their movements are therefore more local, but they are very powerful flyers and can move under their own steam, if you like, great distances over the course of a year. So, for instance, some of the most powerful flyers, like Glossina pallidipes, which is a vector of the trypanosomas causing Rhodesian hat in East and Southern Africa, can move on average about 1 kilometre a day. And over a year, they could move something like 25 kilometers. So the importance of that movement is that fly fronts move. If there's control operations, removing flies, then a lot of that ground can be lost unless it's prevented. So in Iñaki Terrados's presentation, they talk about the problems of reinvasion in relation to ground spraying and aerial spraying operations, where after those operations, the insecticide was not persistent for long periods, and so flies were able to move, and there was nothing to stop them or kill them. So this has been a perennial problem for tsetse control. And using baits, whether it be insecticide-treated cattle, if they are in sufficient density, or artificial baits such as tiny targets, they are really useful in preventing reinvasion and acting as a barrier, and providing that sort of persistent pressure on a population to prevent that movement. So I think that's, in general, that is the problem, a perennial problem we've had with tsetse movement. I think where it is sort of perhaps touches on some of the larger research questions at the moment is, for instance, lots of the Rhodesian hat foci are associated with national parks, with conservation areas in East and Southern Africa. And so questions there arise about how do tsetse, which are infected from infected wild hosts, how do tsetse move out of those conservation areas and into the surrounding farmland, and therefore pose a risk? And how can we control that? And we can't use insecticide-treated cattle in the parks because they're conservation areas. So we have a particular problem of limited tools, and flies constantly, they're not going to largely be controlled within these parks, and they're going to be moving out. So I think that sort of, there's an element of fly movement there, which is a particular research question, and something we as a group at the Liverpool School, working with colleagues in Tanzania and Malawi, are particularly concerned with. And then in the case of, for Gambian hat, where the flies are largely riverine species, again, understanding the movement through river systems. Are tsetse able to cross watersheds? Are they only moving along rivers? And understanding that sort of movement would help us understand better how to deploy targets to prevent reinvasion of cleared areas or rebound as we relax tsetse control.
Thank you very much.
That answers some of the questions.
I think I consider you have answered very well the question. Take advantage to ask Steve for vector control issues. He certainly has answers. But I would say that those preparing the report, there is already there a couple of research questions that were pointed out by Steve that can already enter the list. One is about rhodesiense and the other one about Gambian hat. So thank you, Steve. There's the following question. What can be done with displaced populations? I don't know who I can ask this. This is kind of an epidemiology question. And I don't know who is, do I have a volunteer in the panel? Yes, okay, Steve.
Okay, I can just give you one example from our own experience with displaced populations. So for instance, the large numbers of refugees moving from South Sudan into Uganda had to come from an area with Gambian hat in South Sudan and then moved into nearly 1 million people, I think is the number, moved into northern Uganda, which was an area which itself is also a focus for Gambian hat, but is an area where there's a lot of control currently happening. So Uganda was faced with a particular problem of potentially large numbers of displaced people, potentially perhaps many infected with sleeping sickness, and not only trying to protect them, but also protect the local populations in northern Uganda. And in that case, one of the interventions there was to the Ugandan government implemented tsetse control in areas where the refugees settled to provide immediate protection to people, both those arriving and those already resident. So I think in some of these circumstances, vector control offers a means of protecting people at very short notice, while the sort of clinical interventions like screening and treating people can be implemented.
Good. So let's say that both clinical measures or medical measures can be taken, but as well vector control can both play roles in dealing with the displaced populations. Okay, there's a question: what impact has COVID had on hat elimination? So, who wants to answer? Do you want to answer, Wilfried? Let's see in the DRC if you can comment. The COVID, how did it impact the case finding and the hat elimination?
Merci. Je vais parler lentement pour que la personne qui fait la traduction là, ait le temps de traduire. Effectivement, depuis que Covid est là, déjà c'est l'actualité phare, c'est Covid. Alors, le premier grand impact que Covid a eu, c'est la restriction des mouvements. Alors, ça, comme les équipes mobiles, enfin, les programmes de lutte travaillent avec des équipes mobiles, il y a le dépistage passif et le dépistage actif, mais pour le dépistage actif, cela a été difficile de le réaliser. Donc, normalement, ces équipes mobiles font par équipe mobile 12 itinérances par an. Donc, elles ont 12 fois 20 jours passant d'un village à un autre, faisant les screenings. Mais avec Covid, ces activités ont été sérieusement impactées, sérieusement réduites. Et ça va certainement avoir un impact sur la lutte. Donc, ça c'est une chose. Mais aussi le mouvement des personnes en général qui a été réduit. Et aussi l'attention même, puisqu'en fait, toute l'attention a été focalisée sur Covid et on a oublié du coup d'autres pathologies. Et tout cela a forcément un impact et va certainement avoir un impact sur la maladie du sommeil, sur la lutte contre la maladie du sommeil. Mais tout ce que nous espérons, c'est que cela ne va pas casser la tendance générale. Logiquement, on s'attend à ce que le nombre de cas remonte, mais bon, nous espérons que nous allons continuer cette marche vers l'élimination. Mais ce qui est sûr, c'est que Covid a eu un impact de ce côté-là, de un. De deux, la lutte, comme vous savez, a beaucoup de partenaires, donc il y a beaucoup de bailleurs de fonds qui mettent l'argent pour que ces activités de lutte se fassent, le gouvernement et tous ses partenaires. Mais avec Covid, que ce soit les bailleurs de fonds, que ce soit le gouvernement, a plus dirigé les moyens vers la lutte contre Covid. Et du coup, la lutte contre les autres maladies a été, disons, pas un peu mise de côté. Ça, certainement, ça a eu aussi, et ça continue à avoir un impact sur ces activités de lutte contre la transgéniose africaine. Voilà ce que je peux dire à ce stade. Pour nos activités à nous, des activités de recherche aussi, puisque nous sommes appelés à être assez réguliers sur le terrain et suivre toutes les activités qui se font. Mais il y a eu cette restriction de mouvement qui a quand même impacté sur notre travail. Mais on a trouvé des palliatifs pour maintenir la qualité. Voilà ce que je peux dire à ce stade.
Merci beaucoup, Wilfried. I must say this question goes kind of out of the subject of the meeting, which is about research gaps. However, as Wilfried pointed out, it is also, COVID has been affecting also funding, which may include funding for research and research activities in the field. So we had, before COVID, we have Ebola as well, which was affecting in the same way in countries affected by Ebola. And we may have in the future similar epidemics or pandemics that may be have a negative impact on research as well. So it is, I guess, something to take into consideration, but it's not in itself a research gap that we can point to, in my view. I have Deirdre raising her hand. Do you want to comment, please?
If it would be possible, I'd just like to say a couple of words. So one of the biggest challenges with the impact of COVID on neglected tropical diseases and on HAT in particular is that we've lost our surveillance because the fantastic data that the screening provides was not happening so much during this period. And that means not only could there be transmission going on, but also that we won't see it until there's an opportunity for those screenings to restart. And that's a really big challenge for a lot of neglected tropical diseases. And we don't know exactly how much that is going to be. And while the models can help with that, obviously it's going to be really important to get that data collection back up, as well as the active screening. And I'm going to put in the chat a link to a modelling paper around the potential impact of COVID, which was written now 2 years ago. And one of the side products from COVID is that we'll actually be able to test some of the model predictions around what happens when screening starts. And I think it's an opportunity to learn more about transmission, even within this very tragic circumstances. So I have full respect for the programs going out and trying to restart and get going in these very difficult circumstances. And I think they're going to provide revolutionary information for our understanding of HAT, as well as delivering services to those communities.
Thank you, Deirdre. I also have the raised hand by Philip Busher, if you want to make a comment.
Yeah, well, in fact, I do not agree with the statement of Wilfried that COVID pandemic had an influence on the financial support of HAT elimination and control. I don't think that is true. So please, to argument that statement, I would like to see the data, because I have the impression that there was no cutting short of finances to the HAT control and elimination programs.
Very nice. So this is heating up. We have different positions on some subjects. Thank you, Philippe. Since I have you on the floor, Philippe, there are a couple of questions about PCR. On the you can see, no, you guys, the panelists, you can see all the questions in the questions and answers.
Yeah, there are a lot of questions. I see a question on quel est l'apport de la PCR dans le processus d'élimination de la THA. So what is the impact of PCR on the elimination process of HAT? Well, to answer that question, I think we should first, well, there is definitely a role to play for PCR or qPCR. The thing is that for the moment, there is very little information on the diagnostic performance of PCR in human. What is the specificity? What is the sensitivity? In fact, there is very few information on that. Nevertheless, I think there is, and that is then a research question. I think we should try to have a better idea on the performance of these molecular tests, but not only on human, also on a putative animal reservoir. And I think about domestic animals like pigs, like cattle, etc., but also wild animals. There also the molecular test may play an important role, but again, we should be sure that these tests are really specific, and especially specific for the subspecies that of Brucei that are able to cause human African thrips. So that is a first answer on the first question. Shall I continue?
Yes, because that is, I guess, answering almost the second question. Should PCR be used in routine, like a confirmation test?
Well, I think I wouldn't use it as routine, as a confirmation test in routine, for the simple reason that where the confirmation has to be done, these infrastructures are just not fit to do PCR or qPCR. The confirmation test up to now is a microscopic test. This you can do in a field, in a small health center, in a hospital, etc. But molecular tests, even the most simple ones, like the loop amp, they are very difficult to do in situations where the infrastructure is at low level. But what I think is very important is when we come to a strategy of screening and treating without any confirmation. This is what we expect will be the case with the venue of the new drugs. There, I think it's very important that we could deploy molecular tests for a posteriori testing to see whether the person that was screened and was suspect of being infected and treated, whether that person was really an infected case, a patient. I think that is not only important to economize on the drugs that we are going to administrate to the suspects, but also to know when we have to stop this screen and treat. Because at a certain moment, we will treat probably only suspected people that in fact are not infected. And we have to know sooner or later when can we stop this screen and treat strategy in a given focus.
Okay, I used to call that having a vision, to not to lose vision about the epidemiology in those settings once we start treating all suspect cases and which are unconfirmed. So I want to do my own question over that. Would you say that PCR would be the best, or molecular tests would be the best to confirm in situations like that, given that the other, like trypanolysis or ELISA, are serologic methods that may remain positive for a certain time. But PCR or molecular methods would tell us exactly who was infected and who was not.
Well, again, that depends on the performance, because I can imagine that we have people carrying the parasite, but at undetectable levels for even the molecular tests. Depending on, for example, whether the parasite is hiding in, well, is circulating in the blood or the lymph or even in the CSF. It could hide in the skin. So then it will be very difficult, of course, to make sure that the molecular tests are detecting all the cases. And there, serology, of course, can help. But in that case, again, we should make sure that our serological tests are specific enough not to detect false positives, of course.
Good.
And for example, what I can say already now, if you want to study a putative animal reservoir, forget about the serological tests. There, the specificity is not enough to make sure that if you see, let's say, a sign of an infection in an animal, of an infection with Trypanosoma brucei, whether this is Gambiense or something else.
Okay. Thank you. And I think I continue with you, Philip, because there's a question saying that TB Gambiense was isolated from a dog at a veterinary clinic. And they give us the link of the report about that. And so this is a case of an animal, and they claim it was TB Gambiense. I don't know if you have any comment on this.
That's a difficult one. The RDTs that we have now, they have been developed and evaluated, even validated on human. So we do not have knowledge on how they behave on animals. What I can say from my proper experience, but it was only on one single occasion, when we applied the serocassette, the HAT serocassette of Corey's bioconcept on bovine in Bandundu, or in Kwilu, in Congo. There we found a lot of positives without any other evidence of infection, even not with Trypanosoma brucei. So I would not recommend to use the current RDTs on animals. Okay. And the same applies for the cat test, for example. We know that the cat test, the card agglutination test for trypanosomiasis, which was developed for human use, it does cross-react with other trypanosome infections in animals.
Okay. Yeah, so I was thinking when they say isolation of TB Gambiense that it was not an RDT, but if it was an RDT, we do have the answer. Okay. Something more here, a question by Derek Robinson. What specific lab-based research projects should we focus on and continue to do to fill the gaps in HAT elimination? Diagnosis, although he goes on to ask about insecticides, new treatments, or something else. So maybe we can start by briefly pointing at the research on lab-based projects. What should be the focus? So back to you, Philip. Sorry for the other panelists. You get to answer the other two parts.
I'm sorry. Well, for me, a very important question is, but maybe not for everybody. If we can prove that domestic animals play a role in the transmission into the human population, of Gambians. I'm talking about Gambians and not Rhodesians, because there we know that the wild reservoir is very important. But if we can prove that an animal reservoir, even when it's a very small one, has an effect on the transmission of human sleeping sickness due to Gambiense, then my question is, can we treat our animals for trypanosomiasis in general, and by that way cut the transmission to tsetse flies? And my focus would there be on cattle, but also pigs. We know that we can treat cattle, although the drugs that we have now, I don't think they will be very efficient or curative enough. But what about pigs? We do not know anything about pigs. And I know there is a group in France working on that, and I would be very, very happy to know what they have to tell about.
Okay, but before treating those animals, what about diagnostic or screening techniques? Do we need something on which we should focus now for animals?
I don't think it will be possible with rapid tests. With molecular tests, you can investigate an animal reservoir, but only in a study environment, not on a routine basis. That will be much too expensive. So I think for screening animals, I fear that we don't have a lot of tools, rapid tools, let's say.
So maybe you're pointing to a gap, since we are looking for gaps.
Okay, you can. Okay, go ahead.
So thanks. And then there's the following part of the same question. What about insecticides? Do we need to do some more research about insecticides? This is for Stephen Torr.
Thank you, Gerardo. Can I just sort of come back to also Philippe's, just how he's responded, or several of the questions there. I wonder whether there's also a gap where you talked about, what PCR tests can bring, and the same our understanding of the role of tsetse flies and trypanosomes there. So, and you said one of the problems was that we can't really use these advanced techniques in the field. But is that perhaps a question, is that not a research objective to adapt the sort of techniques are used in Liverpool and Brussels and Paris and Kampala, for instance, and that we could actually apply these in the field in more remote settings, so we could sample livestock locally and tsetse flies. So I think research around that area might be something, I know it's difficult and not possible at the moment, but perhaps that's something that could be achieved in the future. So perhaps just a question perhaps for Philippe to come back to. On the matter of insecticide, we are fortunate in the case of tsetse control that we don't face the same sort of insecticide resistance problems that emerge with mosquito control, where that is a real massive research priority. And we don't really face that with tsetse, probably because part of their population genetics, rates of reproduction, and so forth. And even though we've used just a single pyrethroid for the last pretty much 30, 40 years, I don't think there's any strong evidence of insecticide resistance emerging anywhere. So I don't think there's the same pressing need that there might be for other vector-borne diseases for research on new chemicals. But nonetheless, as new chemistries are developed, knowing whether these are effective against tsetse, so we do have that sort of alternative in the locker. And some of these things are cheaper. And then lastly, sometimes, for instance, there is interest in One Health approaches, where, for instance, insecticide-treated cattle might not only control tsetse flies, but also ticks and mosquitoes. And there, for instance, there are problems. So even though we are treating cattle with insecticides, and we don't think tsetse will develop resistance to them, some of these other vectors will, and certainly do. And so research in that area, in the One Health context, and thinking about the other vectors that we might want to control, especially related to livestock diseases, that would be an area, I think, of important research.
Okay, so rather good news on that front, but some gaps I also pointed out. I will ask Philip to answer to the other question about PCR on tsetse flies, and after we go to Wilfried, who is raising his hand.
Okay. Yeah. Okay. Well, Steve, I would say yes. This is this could be a subject of research, improving, let's say, or simplifying the molecular tests, etc. But I would not put my money in that for human sleeping sickness. I expect more from it from other diseases like COVID, for example. That the other diseases will allow us to follow what is going on the simplification of molecular testing, not only the testing itself but also the sampling. So I would rather say in the domain of sleeping sickness, let's look at the other diseases, what is the development there, and let's piggyback on that research. The little money that is available for human sleeping sickness, I would put it in other research topics.
Thank you, Philip. And Steve, don't give up on this. Now, Wilfried, you are raising your hand. And there is also a question for you, Wilfried, about new treatments. Do we need to...
Ouais, ok, d'accord. Moi, c'était un commentaire peut-être général. Je ne vais pas contredire le professeur Philippe, je suis d'accord avec lui. Et bon, les problèmes aussi dans la maladie du sommeil, pas les problèmes aussi. Basiquement, classiquement, c'est toujours la complexité du diagnostic. Après, bon, c'était la complexité du traitement. Mais sur le traitement, il y a beaucoup d'avancées qui ont été faites et qui continuent à être faites. Mais sur le diagnostic aussi, mais il y a un gap. Professeur Philippe connaît très bien la RDC, il connaît très bien ces milieux très reculés où plus la maladie se fait rare, plus il faut aller très loin, chercher ces malades. Et s'il faut les traiter dans ces milieux-là, il faut que ces petits centres de santé qui sont dans ces milieux très reculés soient capables de diagnostiquer. Or, à l'heure actuelle, les diagnostics les plus sensibles, enfin parasitologiques, ça reste la mini-colonne, mais qui est difficile, voire impossible à faire au niveau de ces postes de santé-là. Donc, il serait intéressant, continuer à explorer d'autres pistes, comment simplifier les tests moléculaires ou comment, je ne sais pas moi, simplifier la trypanolyse, de sorte que, bon, on arrive à des tests qui soient plus ou moins sûrs, mais dans ces milieux très reculés, pour qu'on soit à même de traiter, parce que sous les traitements, on a fait beaucoup d'avancées, mais pour éliminer, il faut vraiment aller jusque-là. Il faut élargir la couverture par ce dépistage passif et par ce traitement. Ça, c'était juste un commentaire général. Alors, la question pour moi, c'était laquelle, Gérardo?
Je crois que tu as répondu à la question. C'était s'il fallait investir aujourd'hui dans de nouveaux traitements, dans de la recherche pour de nouveaux traitements.
La traduction ne marche pas.
Mais tu as répondu à la question, en fait. I wanted to comment on...
Ah, okay. I've already answered. Okay, good. So thank you.
I wanted to comment on what Wilfried just said about the necessity to be able to screen and test individuals that would be good to treat in the future, especially thinking of the new medicine that is coming, that is looking good, called acosiborol, that could change all the rules about who we are going to treat. So I wanted to just, as an information, to say that in the WHO, we have been working with experts on screening and diagnostics to develop a target product profile, a TPP, that sets the parameters and what we would like to have as a test to identify individuals that should receive treatment. It will be a test that can be used in peripheral settings, ideally, and that can raise the level of suspicion on individuals so that we are more sure who requires treatment in the future. So that TPP is out for any research groups that may, or funders who may want to invest on that, on looking for those tests. And this is what Wilfried was pointing out as a gap to fill. Okay. So what do we have next? What may be the effect of climate change on heart transmission and geographic extent? Okay, so that question I would orient towards Steve, because climate change obviously will affect tsetse flies, but maybe not only, is affecting already all of us humans as well. But please, Steve, if you can comment.
Okay, thank you. Yes, I think that's an excellent question. And you know, already there is evidence of climate change and also just the impact of anthropogenic change on the environment, having knock-on effects on sleeping sickness right across Africa. So just to give you a couple of examples, work that we've done in Zimbabwe, there gives pretty strong evidence that increasing temperatures in southern Africa have had an impact on tsetse populations, and in some of the, if you like, traditional hat foci in the Zambezi Valley, numbers of tsetse have probably declined by 2 orders of magnitude over the last 30 years. And but as hot places got hotter and, if you like, had an impact and limits how tsetse populations can grow, some of the cooler places have become warmer and now probably have become suitable for tsetse. And, you know, there's a little bit of, you know, perhaps it's circumstances, but would also fit with this, how the cases in Zimbabwe in recent years have been in the higher areas around Makuti, in the escarpment above the Zambezi Valley. So perhaps evidence of impact of climate change. So I think that tells us that climate change is going to shift the distribution of cases, of tsetse cases. You can also see perhaps signs of this in West Africa, work of Fabrice Cortin and others from IRD, looking in places where historically there were Gambian hat foci, and those seem to have either gone quiet or disappeared. Tsetse is still there, but there's been large anthropogenic changes, which have possibly changed the sort of dynamics of the tsetse in the human populations. And I think, as you alluded to at the beginning of the question, Gerardo, is we know that climate will affect insects and tsetse flies, but it will also have impact on livestock population and human populations. And so they too will be moving. So I think it's a really big, you know, that is a massive research question for the future, trying to predict what will happen, where we should be looking. You know, diseases may be in new places, and old diseases and old foci may actually go quiet, and not because of something that we've done intentionally, but a sort of incidental consequence of anthropogenic change.
Thank you. I want to ask, Deirdre, I was about to ask you your opinion.
No, I think everything that Steve has been saying is really interesting, and I think it feeds back into the discussion around diagnostics. I mean, maybe the diagnostics that we would like to use to really stamp out hat are not necessarily the diagnostics that we might use in order to do surveillance of new foci. Obviously, we'd like the best possible diagnostic always in the best place, but less perfect diagnostics that could be rolled out for surveillance in places that we think may be at risk may be a useful tool. So thinking about that population level screening, followed up by, you know, more intense screening at health centres when people come in for other diseases, you know, thinking about the tools that we can use to identify new foci, I think is an interesting area of future research.
Thank you. So if I interpreted well the comments by Steve, the climate change is going to make flies move to a new location, and humans also are going to be moving in the same locations, looking for basically for less hot places. So it is looking like almost nothing will change, I would say. But of course, this is much more complicated, and Steve was touching on that. Many other factors may be involved in the responses or the adaptations to climate change. So very interesting for modelers and so. Okay, I want to jump a question about taxonomy, because I don't think that leads to gaps in research, really. There's one. This one about from Rachel Bronson. It was kind of already answered. What are the biggest challenges in assessing the potential role of animal reservoirs in Gambian Sahara? What are the key research questions today? What tools are needed, but not yet available to answer those questions? So we were already going into that field, but I want to open. I don't know if Philippe has any additional comment about these questions.
Well, I think for investigating an animal reservoir, what we need is more specific. More specific, but also more sensitive tests to detect Trypanosoma brucei gambiense and even rhodesiense. Mm-hmm. The more sensitive molecular tests, they do detect Trypanozoon, but that means that they will also cross-react with brucei brucei, which is not infective to human, and that occurs in animals. And this is something that at ITM, but not only at ITM, that we investigate what new molecular tests we can develop that are more sensitive in detecting Gambiense in animals. That is the main question for the moment. And we can do, of course, two steps. You can first do a molecular testing on an animal to see whether there is Trypanosoma brucei sensulato. And then, if that is positive, you can try to confirm that this is Gambiense or not, or rhodesiense or not. So two steps are possible. And the first step for me could be, let's say, an argument to treat any animal that we found positive for brucei sensulato. But if we had more sensitive Gambiense test, molecular Gambiense test, I would be very, very happy. And for the moment, they are lacking. Not to say that there are not candidates. There are several candidates in the pipeline, but we should validate them, which is not yet done.
Okay, thank you. I think that gives us a good answer to a situation today. All right. There's a question back to vector control. If the technique on spraying animals' stomach and leg area only, might that encourage development of tick resistance to the chemicals used? Maybe Steve can say something.
Thank you. I think that's the general question about applying insecticides that will have an impact on ticks. Whether it's what they're describing is a restricted application of pyrethroids, which is tsetse flies concentrate on the legs and belly, and you could reduce the costs and the amount of insecticide used by focusing there. And some ticks also are concentrated in these areas when they're migrating onto the animal. But also other tick species are, for instance, in East Africa, the brown ear tick is on, as the name suggests, on the ears, and that method will completely miss most of the ticks, these brown ear ticks, which are major vectors of East Coast fever. I think what you can do is you can adjust the application to try and reduce either targets particular species or reduce their impact. So the restricted application technique also reduces not only the amount, but where we put it and how frequently we apply it, with the idea of, if we want to, allowing livestock to develop enzootic stability against diseases such as babesiosis, cowdriosis, and so forth. So yes, it is a problem, but I think we can sort of manage. This is really, working with people working on tick-borne diseases. How do we control ticks without leading to acaricide resistance?
Fine, thank you. There's another question kind of related, because it's a one called coming from Delphine Otteman. She's saying research on diagnostic and new treatments have been mentioned, but what about research on a vaccine for cattle to control TB rhodesiense and maybe TB gambiense? So a vaccine for cattle. Anyone would have a comment to make? So Deirdre, please.
I'm afraid I have nothing to say about vaccine development per se for HAT, but I think it's striking that the two diseases which have been eliminated have been vaccine eliminatable, vaccine controllable. And I think it's important to consider the difference between eliminating without a vaccine and with a vaccine. Smallpox and even polio, which has a vaccine, which is quite effective, is still struggling to eliminate. And the whole of the rest of the world has to keep vaccinating while those problems are sorted out. And so it's no small task getting to this final mile. And we know that as we press diseases down, they become much more heterogeneous, which is what we're seeing also with HAT. So we see different behaviour in different places in terms of the dynamics, but also it's a social problem, which, I'm glad you're talking about that tomorrow in the next meeting. So just to note that eliminating without a vaccine is a very challenging thing, and maybe this question isn't so ridiculous in that context.
Thank you. Is there any other reaction in the panel? Okay, if not, I guess we can say for eliminating this disease, at least the Gambians we are lucky that this is a disease vulnerable to other interventions that we have, and we could almost say we don't need a vaccine at this stage. Of course, if we had a vaccine, we would probably use it anyways. Okay, I'm running out of questions actually from the participants. I think the question that remains that I see there is the one from Julien. What is the role of PCR in the process of elimination of HAT? We did cover about PCR already, but maybe there is more to say specifically if we want to frame a role for PCR in the elimination process. So, and I see Philippe is raising his hand, so please, Philippe.
Yeah, but not to answer that question, just to complement of what I proposed as research topics in answer to Rachel's questions. What I think is also important is to investigate improved methods for sampling specimens for molecular analysis. We all believe that for detecting a Gambiense infection, and it may be also the case for rhodesiense infection, if we want to make them very specific, we for the moment are targeting single or few copy genes in the whole genome. And we have evidence, at least from my team, we have evidence that detecting RNA is much more sensitive than detecting DNA. So I would recommend to investigate improved sampling methods to stabilize RNA in the harsh conditions that prevail in sleeping sickness foci, to stabilize the RNA so that you can transport it to a reference center for analysis of RNA, specific RNA for Gambians or Rhodesians.
Thank you. Because, Philip, RNA needs to be transported at -20 degrees or something like that?
Well, for the moment, it's best to keep it frozen. Well, to first stabilize it in a chemical and then keep it frozen, also during transport. You can keep it for a couple of days or maybe even a week at a lower temperature, let's say what we call ambient temperature in Europe. But if you face 35 or 40 degrees in Africa, in hot foci, then there is a serious problem with RNA. So if we can find ways to simplify and the collection methods and to improve the stability of RNA, that would be a great leap forward, I think.
Good, and you are already at least partially answering that question, because if we had better transportation methods, sampling and transportation, the role of PCR in the elimination would already grow.
Sure, yeah.
Thank you. So I see some new questions coming up. So back to vaccines. Is this vaccine approach worth considering for HAT? And then it continues, an invariant trypanosoma vivax vaccine antigen induces protective immunity. Something that was published in Nature 2021, recent article. Okay, is there any reaction to this? We already were talking about vaccines. Is it worth considering? Could it be pointed out as a gap? We were kind of saying no. Yeah, Wilfried, he wants to.
Bon. Je pense que Gerardo l'avait déjà dit. Hein. Bon, de mon point de vue, non. Et aussi, étant donné le nombre de personnes qui sont touchées, bon, voilà, c'est non, je ne pense pas que ce soit nécessaire. Mais je voulais mettre aussi sur la discussion, mais un autre volet, mais toujours de la marche vers l'élimination. C'est le dépistage actif versus le dépistage passif, puisque là, ça fait aussi sujet à beaucoup de discussions. Je pense que ce soit le prof Philippe ou Gerardo lui-même est au courant de tout cela, puisque nous avons les équipes mobiles qui font du dépistage actif et qui sont de plus en plus coûteux. Ça fait que pour avoir un malade, cela coûte de plus en plus cher. Mais de l'autre côté, nous avons le dépistage actif qui aide à élargir, disons, la couverture, puisque le taux de couverture des équipes mobiles n'est pas très grand par rapport à la RDC. Bon, au moins, tous les foyers sont plus ou moins visités, mais avec le dépistage actif, on sait aller très loin. Mais les deux ont quand même des limites, puisque, bon, il y a beaucoup de discussions maintenant sur le fait qu'est-ce qu'il faut maintenir les équipes mobiles alors que nous allons vers l'élimination. Elles sont de plus en plus coûteuses et avoir un malade, ça coûte de plus en plus cher. Est-ce qu'on les laisse tomber et on fonce sur les dépistages passifs qui demandent aussi beaucoup d'informations, mais surtout en termes de diagnostic et des moyens faciles. Je n'ai pas une solution tranchée, mais c'est une question que je mets aussi sur la table de discussion.
Merci, Wilfried. Is there any reaction of the other panelists to this? But it was put on the table of discussion, so perhaps it can be pointed out as another gap. Deirdre, you're raising your hand.
So I think the question of to what extent active screening can be rolled back and you can rely on passive screening, and when you might be able to do that is a really interesting question. And I know that there's a number of modelers on this call who have already started thinking about that. So if they wanted to post anything in the chat about their thoughts on that. But I think that's going to be an important logistical consideration for the coming years.
Thank you. I see another question from Heather talking about RNA, but I think this was already explained. She's wondering what about using FTA cards, blood on filter paper of FTA cards. And I think it was explained that what we have a limitation on temperature. The RNA, if I understood well, would not stay. It would not work on simple filter paper or FTA cards. But maybe Philip can jump in.
Yeah, apart from the stability of RNA, there is also the volume of the sample. Like even on FTA cards, the volume of the sample that you can put and from which you can extract the DNA or the RNA is very limited. And this is why we should try to find better methods to collect larger samples from which then we eventually can isolate RNA or DNA. So blood on filter paper or FTA cards, for me, that is not sufficient. We should find larger volume collection devices, and they exist. They exist.
Mm-hmm. Yes, for DNA, no, those sampling methods are useful for DNA, but we have a different problem with DNA.
Yeah, but also for RNA, they do exist. But there, then the stability is another issue. Yeah. Mm-hmm. So two issues or two characteristics of a sampling device, an alternative sampling device: stability of RNA and increased volume of sample.
Yeah. All right. Thank you, Philippe. And then this last question I see is probably going back to you, Philippe. Can we envision validating a test of cure?
Of course, you can do that, but you can do it only in clinical studies. I fear that the number of patients that are treated for the moment in the whole of Africa is not sufficient for proper validation if it's not in the framework of a clinical study. And this is why, for example, together with DNDi, several groups are indeed proposing validation of the new molecular tests that have been developed at different research institutes. For serology, I don't think we have good candidates to be validated as test of cure, for the simple reason that the only serological tests that really work are antibody detection tests, and antibodies, they stay for a very, very long time in the circulation, even after cure.
Good. And I add to the question, because okay, not enough patients right now to plan such a study, but what about using retrospective data banks, sample banks? I saw recently there was a publication using the samples from Bujimaji that had a good follow-up. Could that help be used for validating a test of cure? It's probably, you tried, probably, I don't know.
Well, that was recently done with for the antibody detection tests, and there we confirmed that antibody detection tests do not work for that. That was a study published by the group of Raquel, and then for the rest, in Bujimai, we collected samples that can hardly be used for validating molecular tests. But again, in two things. First of all, these samples are not fit for RNA analysis. And secondly, in that particular study, what we saw is that the molecular tests that we applied on the blood and the CSF of patients before and after treatment, the molecular test that was DNA detection was still positive in a fraction of persons or patients that were considered cured by all other means, by all other parameters. After 2 years, they were considered cured, and some of them were still positive in for DNA, trypanosome-specific DNA in the CSF. And that was about, if I remember well, 20%. So 1/5 of the cured patients were still positive, but only in the CSF. Yeah. So if we concentrate on blood, we may have more chance to find a test of cure. But I would go for RNA detection in the framework of clinical studies, which means that for routine, probably you will not be able to to use them on a large scale.
Thank you.
I hope I don't sound too pessimistic, yeah?
We need to be realistic.
Okay.
Okay, thank you. There's a new question, this regarding vector control. And maybe I will read it. It's saying, in Kenya, we are promoting the installation of livestock protective fences. It's called LPF, or the zero fly netting on zero grazing units to control tsetse flies and other biting flies. There has been research that has shown that the netting also reduces mosquitoes in households with the nettings installed on their zero grazing units. What are the contributions of use of the nettings on HAT control in the countries that have the HAT problem? And this goes obviously to Steve.
Thank you. So thinking first, Kenya and Rhodesian hat and countries affected by that. So in East and Southern Africa, I think they're, effectively treating cattle with insecticide, which is what this is doing. It means the cattle are protected from flies, or flies trying to feed on them are going to contact the netting and be killed by the insecticide. So that really works in grazing systems, zero grazing systems, which are common in some parts of East Africa, but not in the sort of typical HAT foci of say, around the Serengeti and Tarangire parks. It's more in coastal Tanzania and coastal Kenya. So in particular situations, yes, it could contribute something towards transmission of trypanosomes, including trypanosomes affecting humans, but I don't think that there are many of these sorts of livestock production systems in areas where there's sleeping sickness in East and Southern Africa. I think that approach might have some utility. So I think as Philippe mentioned earlier about pigs may have a potential role. And certainly I've seen in Central and West Africa, large piggeries and with sustaining tsetse populations, you can see lots of flies in some of the foci in DRC with Quonsensis feeding on pigs. And perhaps that's a way to, if the research that Philippe and others are doing on pigs perhaps being reservoir hosts, then that might be a way of controlling tsetse flies using that and interrupting that particular route between flies and pigs. And just generally on netting, but we think that part of the sort of where people are possibly infected is when tsetse flies come into houses during the hot parts of the season, into huts, and then they're resting there trying to basically find a slightly cooler place, and then they become trapped inside with humans. And normally lots of these flies in Eastern and Southern Africa do not naturally feed on humans, though it seems bizarre to say so, given that they obviously do at some point. But it seems to be in particular situations, and possibly when it's very hot, flies come into houses get trapped and bite humans inside. So if you had, for instance, insecticide-treated netting on windows just to protect and reduce that invasion of houses, that might be a particular way of protecting people. So I think it might have some utility in particular circumstances. And just generally, of course, if these sorts of interventions that farmers do to protect their cattle, farmers are largely paying for because they want to have more productive animals. And it's a sort of speaks to a One Health approach of where you harness the resources and aims of livestock keepers and try and use that to help a human health problem. Can I just, while I have the floor,
okay,
can I just, I thought Deirdre made a really nice and important point actually when she raised the point about vaccines and saying that is, just remember, none of these diseases have been eliminated that we didn't have a vaccine for. And then right at the beginning, Roy also reminded us that only 2 diseases have ever been eliminated. And so we are, this sort of really says this is a really big challenge to me. We're doing tremendously well as a community, but to reach that elimination, that global elimination goal is a massive ambition. And we hope we can do it. And then Deirdre has highlighted, we haven't got one really important tool probably. And in the circumstances, we haven't got the numbers of cases to do the research, to even test these things. So it's a really difficult problem. And then lastly, I'm also reflecting on, there's not a strong evidence of people raising a strong immune response and becoming immune. So probably the chances of getting a vaccine are pretty slim in all those circumstances. So if Deirdre has reminded us how powerful that tool is and we haven't got it, I think that also reminds us, look at our current tools, what we have got available, and what are we going to do in the next 10 years? So how are we, some of the things we've already mentioned about monitoring, how are we going to do that? What are we going to do when we find it? How do we do reactive treatments, reactive control, and so on? And, there's a big task ahead of us, and we've got possibly one hand tied behind our back. So personally, I think we can do it, but I think it's a real good reminder from Deirdre, the scale of the problem. Sorry.
Yes, Deirdre, go ahead.
I'm sorry for slightly hijacking your conversation, but I think this is also tied to Wilfred's point about scaling back active screening. I think we really need to think about what is the alternative surveillance method, and Philippe made some very good points about serology, and they're really disappointing points. But, what does the future look like for passive surveillance of HAT when numbers are really low? And then also Rachel's made a good point in the chat about using maybe passive xenomonitoring. So I think there's a huge research area about what is going to happen when the cases get low. We can't measure zero, that's way too expensive. And, and also Wilfred's point, if you don't mind me also saying about funding, funding may have been maintained now, but as numbers get really low, the investment in monitoring and evaluation needs to really step up. And that's a big ask from donors. So I think these are the challenges which are very important, and Steve has also highlighted them.
Thank you. Jumping on what Steve had said about that, it would be the only disease eliminated or eradicated, maybe, where there is no vaccine, without vaccine. So that constitutes in itself maybe a carrot for institutions and donors to follow. It would be contributing to this major achievement of humanity. So it can be somehow encouraging. Also commenting on what Steve was saying about protective fences and pigs. If I'm not mistaken, I think pigs always come number one. All the studies have looked for TB Gambians in animals. There's always pigs as number one. And if they can benefit from protective fences technique, that could have, I think, an impact. I want to jump on Rachel's question because I think we already answered that question about monitoring the presence of TB Gambians in insects. We have a big limitation there about the techniques, the PCR, that we don't have good enough techniques to do xeno monitoring. And I think that applies not only to tsetse or to other infecting or other insects that could have blood meals from humans. But maybe there is another. Okay, Deirdre, you want to react? Go ahead.
And again, but also I think that it's really important to test these techniques now while we know there are some HAT cases. We don't want to start testing these techniques when we think there are no HAT cases. They need to be validated when we think there are HAT cases. So maybe we need to push the boundaries of some of these tools now and test them in the field, because when we don't see HAT cases, it's going to be very hard to test what a zero means on maybe a xenodiagnostic surveillance site. So, maybe this is an area again where lab research, could try to make those more sensitive or specific.
Okay, although we will still be able to infect flies, even if there are no more cases to carry out this type of research.
Oh, sorry, my understanding of this question was if you just took samples of mosquitoes that were biting humans and looked to see if they had Gambian, so doing that sort of passive surveillance, but maybe that was misunderstanding on my part.
Okay, we have one new question. If we can look at this, is in French. So, okay, it was strange that nobody had asked about trypanosomes in the skin. So with the identification of the skin, like a new site for trypanosomes, do you think that considering this reservoir, the dermal reservoir, the diagnostic strategies, as a diagnostic strategy, we can easily reach elimination or even avoid re-emergence of HAT and animal also, animal trypanosomiasis. So I guess, Philippe, you are on the first line to say something about this.
Thank you. Interesting question, difficult to answer. First of all, I'm not convinced that that many patients bear trypanosomes only in the skin and not in other compartments of the body. So this is something that I haven't seen enough data to be sure of it, that a person with trypanosomes or an animal with trypanosomes only in the skin cannot be detected otherwise, even with serology, as a suggestion. This for me is missing. The evidence is not strong enough. But okay, putting aside this this question, if it is true that persons carrying, that there are sufficient persons carrying the trypanosomes only in the skin and not detectable in other body compartments, the question remains, how are we going to transform this evidence into a test that allows us to use this as a diagnostic test that is then followed by a treatment. Skin biopsy in itself, it is invasive, yeah. Then how many trypanosomes are there in the skin? Is that sufficient to have a sensitive detection of the parasite in that sample? These are open questions to me. I cannot answer because I haven't done any study on that. But the evidence that is out, for me, yeah, it's not sufficient to convince me that it is an important diagnostic specimen. And if it is, then we have to to develop devices for skin biopsy that is acceptable for the person and that is collecting enough material to do proper diagnosis.
Thank you, Philippe. I think that's a good answer to the question. We are not quite ready to envision this. Some of the data still needs to be coming, and also the possibility of having a field applicable test. Any other of the panelists want to?
Oh, I just want to add to that, that is a subject of study that is going on within collaboration with the NDI. There is a sub-study that is going on, and I hope that some answers will come from that study. Let's hope.
Okay. Thank you. I don't see any more questions coming, and we only have 10 minutes left. I guess I can open for any other additional comments any of the panelists would like to make. And if not, we can close the meeting. I think we have gathered a list already of gaps that were pointed out. Maybe Deirdre, you want to jump in? Yeah, thanks.
Yeah, I just want to plug maybe tomorrow's meeting, because as you know, you highlighted the modelling study on vexenidazole, which showed that if you increase uptake but decrease compliance, you may end up with more of a problem. And I think it's really important as we move down to these final foci that we have an understanding of the social drivers of uptake and of all of these different interventions. So I think there's going to be some fascinating stuff tomorrow, and I'm looking forward to it.
Yes, thank you. I agree with that as well. So that makes me remind all the participants of today that to participate tomorrow and after tomorrow in the same afternoon meetings, you have to register. So don't forget to register to be tomorrow here. All right, so I guess it's back to the organizers. I will consider the discussion finished, and maybe you have other remarks to make before we all jump out. And before that, I want to not forget to thank all the panelists that participated. You have given your time, and I think we have a high-level panel today. We have very good questions and reactions to the questions, and that provided us deeper knowledge about where the gaps are, where the challenges are in terms of research, particularly. So thank you to you all. Back to
Gerardo. Yes, Gerardo, thank you very much indeed. And thank you to all the panelists. Their most interesting discussion. I think there were at least 3 or 4 important markers for future research, which we're returned to on the final day. But just to encourage everybody to continue, as Deirdre mentioned, there's a session, interesting session tomorrow, and we look forward to furthering discussions. So again, thank you, Gerardo, very much indeed for chairing such a great meeting. Thank you. Look forward to seeing you tomorrow.